Hello, my name is Avril Coghlan. I am a bioinformatician in the Sanger Institute in Nick Thompson's group, and I am going to tell you about Vibrio cholerae genomic data and how you can analyse it using the Pathogen.watch website. First, I will give you a brief overview of my talk. I will introduce the Vibrio cholerae reference genome and talk about what other genomic data is available for Vibrio cholerae. I will introduce Pathogen.watch and its Vibrio cholerae component Vibrio Watch. I will explain analyses that you can carry out on Vibrio Watch on your genomes that you upload, as well as the genomes already uploaded by us. I can explain how to upload a new genome, and I will lastly mention some future plans that we have to improve Vibrio Watch. The Vibrio cholerae genome was first sequenced by Heidelberg et al. In 2000. They sequenced an L-Tor strain N16961. They found that the genome is 4 megabase and consists of 2 circular chromosomes, a larger chromosome of about 3 megabase and a smaller chromosome of about 1.1 megabase. Together, these 2 chromosomes have about 4,000 genes, and their GC content of the chromosomes is about 47%. It's worth bearing in mind that N16961 is the reference strain, but there may be a wide variety of genomic variation in the Vibrio cholerae population worldwide. Some interesting examples have already been reported. Johnson et al. In 2015 reported a non-O1 isolate that has the 2 chromosomes fused into one big chromosome. Okada et al. In 2015 reported an O1 L-Tor isolate with a 3rd replicon of nearly 1 megabase, so that its genome size was 5 megabase. As we sequence more and more genomes of Vibrio cholerae isolates, we are likely to see more and more genomic variation. Since the year 2000, genome sequencing technologies have greatly improved, and this has meant that many different Vibrio cholerae isolates have had their genomes sequenced. An example of this was a key paper by Chun et al. In 2009, who sequenced the genomes of 23 diverse Vibrio cholerae isolates that had been collected over nearly 100 years. These isolates included both clinical and environmental isolates from many different countries. In this phylogenetic tree of the 23 isolates, you can see that the isolates causing the current global pandemic, or 7 pandemic, are all belonging to a particular clade, highlighted with the red box here. These 7 pandemic isolates belong to a larger clade which has been named the Phylocore Group 1 clade, or PG1 clade, and which has a sister clade called the Phylocore Group 2 clade, or PG2 clade. The Phylocore Group clades are distantly related to the other isolates sequenced, which are also all distantly related to each other. On this slide, I show a plot with the number of isolates sequenced in 22 key papers, ranging from the year 2000 at the top to 2022 at the bottom. Together, these 22 key papers have published 3,254 Vibrio cholerae genomes, of which 84% belonged to the 7 pandemic lineage. All of this genome data has been useful to epidemiologists to try and understand the spread of Vibrio cholerae around the world, and also to scientists and epidemiologists interested in Vibrio cholerae genome evolution, for example, the spread of antibiotic resistance genes and the presence or absence of virulence genes. Of course, these 22 key papers do not include all papers that have published Vibrio cholerae genomes. In fact, if you look at the NCBI or ENA databases, you will be able to find about 4,250 Vibrio cholerae genome assemblies. These will include some genomes that were published in papers that I have not listed here, but also some genomes that have not yet been published. Furthermore, if you look in the ENA database, you will see about 13,400 Vibrio cholerae sequence runs, which are unassembled reads for Vibrio cholerae isolates. This reflects the fact that many researchers in the past have not assembled reads into assemblies, but simply aligned the reads to the reference genome. Clearly, there is a huge amount of genomic data available for Vibrio cholerae, and my goal as part of the Vibrio Watch project is to make all this data available and useful for epidemiologists and researchers studying Vibrio cholerae. Next, I will tell you about Pathogen.watch and its Vibrio cholerae component, which we call Vibrio Watch. Pathogen.watch is a website that can be used as a global platform for genomic surveillance of pathogens of humans. It has been developed by the Centre for Genomic Pathogen Surveillance, a collaboration between the University of Oxford and the Wellcome Institute. So far, Pathogen.watch includes almost 76,000 genomes for bacterial pathogens of humans that cause worldwide problems. I have shown here a phylogenetic tree of some major bacterial pathogens of humans, showing the number of genomes that are in Pathogen.watch so far for those species. You can see that there are 4 species that comprise 83% of the genomes added so far. This reflects the fact that the research community studying those 4 species have found Pathogen.watch to be an extremely useful website for disseminating and analysing genome data for their species. We are also convinced that Pathogen.watch will be an extremely useful website for Vibrio cholerae, and so we have started to upload Vibrio cholerae genomes to Pathogen.watch. So far, we have put 390 genomes on Pathogen.watch, but as I mentioned on the previous slide, there are many thousand of genomes already available, and our goal is to upload as many as possible in the next year. If you go to the Pathogen.watch website, you will be able to see a map of where the 390 Vibrio cholerae isolates whose genomes are currently in Pathogen.watch were collected. For example, you can see 92 were collected from Mexico, 34 from Haiti, and 59 from China, and so on. You can also see a timeline showing the dates when these isolates were collected, which range from the 1930s to around 2012. As we put more and more genomes into Pathogen.watch for Vibrio cholerae, these maps and timelines will be updated. Next, I will explain some analyses that can be carried out on the Vibrio Watch website, both of the 390 genomes already uploaded, but also of genomes that you upload yourself into your private space in the Vibrio Watch website. One of the simple analyses that Vibrio Watch provides is some statistics on the assemblies. This can be useful to identify anomalous assemblies that may be contaminated or may represent very diverged Vibrio cholerae. For example, on the left I show a plot with, on the y-axis, the size of the assembly, and along the x-axis all of the Vibrio cholerae isolates. You can see that the majority of isolates have a genome size of about 4 megabase, around the size of the reference genome. However, about 4 of the isolates indicated with arrows have very unusual genome sizes. For example, isolate HE45 has a genome size of about 6.5 megabase. And if we look at other plots on the Vibrio Watch website, shown on the right, we can see it has an unusually high number of contigs and an unusually high GC content. In fact, when I analyze this genome using an external software, CheckM, to find potential contamination of other species, I found that this genome is approximately 60% contaminated by DNA of other species, which presumably explains its very large assembly size. Contamination also seems to explain the strange assembly size of PCS022 and 1551CDC. However, the fourth genome, CT536993, has a very small genome size of 3.5 megabase, but does not have an unusual number of contigs or GC content. This isolate was collected from sewage in Brazil and is a non-O1 isolate, so is distantly related to the 7 pandemic lineage. This makes one wonder, maybe it has truly a very small genome of 3.5 megabase and represents a genomic variation from the reference isolate. One of the most useful type of analyses that you can carry out using Vibrio Watch is to try to use it to figure out how isolates are related to each other. One method of doing this is to use multi-locus sequence typing information, known as MLST. In Vibrio Watch, we have implemented the MLST scheme of Octavia et al. From 2013, a commonly used MLST scheme for Vibrio cholerae. This scheme is based on 7 housekeeping genes, and if 2 isolates have identical sequences at those 7 genes, they will have the same sequence type, while if they have very different sequences at the 7 genes, they will have a different sequence type. The 7 pandemic isolates are nearly always sequence type 69 in the Octavia scheme. If we look at a tree I've made here of all the Vibrio cholerae that are in Vibrio Watch so far, you can see that the 7 pandemic isolates form a clade shown with the red bar, and that almost all of these are sequence type 69. There are also a couple of clades of non-7 pandemic isolates indicated with the green bars. When I make a tree just of those non-7 pandemic lineage isolates and plot beside the nodes of the tree the sequence type number in the Octavia scheme, and also put adjacent to that the metadata for the isolates, namely where were they collected in the world, and also what type of isolate it was, either clinical or environmental or so on, and also the serogroup, if available, I can see in this tree that there are some groups of closely related isolates that have the same sequence type in the Octavia scheme. For example, highlighted with the red box is a group of closely related isolates that all have sequence type 737, and from the metadata we can see that these are all clinical isolates that were collected in Mexico. Since these are non-7 pandemic lineage isolates, they must represent isolates collected from endemic Vibrio cholerae disease in Mexico. Since the MLST scheme of Octavia et al. Was based on just 7 genes, more recently Liang et al. In 2020 have created a core genome MLST scheme for Vibrio cholerae. This is based on 2,443 core genes, that is, genes that are present in almost all Vibrio cholerae isolates sequenced so far. In Vibrio Watch, we use this core genome MLST scheme to place isolates into clusters of closely related isolates. For example, one of the isolates that I have looked at, HCUF01, is an isolate collected in Haiti in 2010. In Vibrio Watch, it is shown to have the sequence type 69 in the Octavia scheme, so is a 7 pandemic clade isolate. Vibrio Watch shows a core genome MLST cluster containing the isolate HCUF01, which I've shown on the left here. This shows that HCUF01 is closely related to other isolates from Haiti, shown with green circles, but is also closely related to some other isolates from Nepal, shown with red circles. This suggests that isolates from Haiti are closely related to isolates from Nepal. This agrees with analyses that have been published on the origin of the Haiti epidemic in 2010, which led to the hypothesis that Vibrio cholerae was carried from Nepal to Haiti. Another very interesting analysis that you can carry out using Vibrio Watch is to check for the presence of virulence genes. It is well described that certain virulence genes can make the pathogen more virulent, causing more severe disease. The most important virulence genes for Vibrio cholerae are the CTXA and CTXB genes, which encode the cholera toxin, and also the TCPA gene, which encodes the toxin co-regulated pilus, which is important for colonization of the host. There are also some other virulence genes of slightly less importance, such as ZOT, ACE, and so on. On the left, I have made a tree in Vibrio Watch showing the presence of the CTXA gene as red nodes in the tree. You can see that it is present in all of the isolates in the 7 pandemic lineage, indicated with a blue bar, but it is also sometimes present in non-7 pandemic isolates, which are indicated with green bars. On the right hand side, I have made a tree of just the non-7 pandemic Vibrio cholerae that are in Vibrio Watch, showing the CTXA gene as red node in the tree again. Here we can see that all of the isolates that have the CTXA gene are actually all belonging to one clade. In fact, this clade is the phylochore group, or PG clade, which I mentioned previously, a clade of isolates that are closely related to the 7 pandemic lineage. This tells us that the CTXA gene is limited mainly to 7 pandemic isolates, but also to some closely related isolates. This type of analysis can be useful in figuring out the likely virulence of an isolate that you have sequenced. A key use of genomic data for epidemiologists is to predict antibiotic resistance based on the genome. Based on this information, it is possible to predict which antibiotics the Vibrio cholerae causing a particular outbreak or epidemic are likely to be susceptible to and which they are likely to be resistant to. In Vibrio Watch, we predict antibiotic resistance based on looking for antibiotic resistance genes and variants in them. And so far we can predict resistance to many different antibiotics such as aminoglycosides, chloramphenicol, and so on. For example, I have shown here a tree of all the isolates in Vibrio Watch from Haiti, with the red nodes in the tree showing isolates that are predicted to be resistant to chloramphenicol. You can see that when I show with the blue line the 7 pandemic isolates, all of the 7 pandemic isolates are predicted to be resistant to chloramphenicol, and all of the non-7 pandemic isolates are not resistant, that is, they are susceptible. And this is actually in agreement with the phenotypic data on antibiotic resistance for the Haiti isolates. Of course, although these 7 pandemic isolates that caused the Haiti epidemic were resistant to chloramphenicol, fortunately they were susceptible to tetracycline and doxycycline. I will now explain how you can upload a genome that you have sequenced yourself or several genomes to Vibrio Watch to analyse it. On the Vibrio Watch website, you can make a private account where you can upload genomes that you have sequenced or any genomes that are not presently in Vibrio Watch. These data are kept private to your own Vibrio Watch account, as are all the analyses that you produce on those genomes. Nobody else will be able to see either your genomes or your analyses. You can carry out all the analyses that I have described so far, including MLST analysis, finding close relatives using core genome MLST, finding virulence genes, predicting antibiotic resistance, producing phylogenetic trees, and so on. Here I show an example of an isolate that I uploaded the genome for, isolate VC228305, which was collected in 2005 from an outbreak in Argentina. When I uploaded it into Vibrio Watch identified it as sequence type 170 in the Octavia scheme, so it is not in the 7 pandemic lineage. I then made a tree of all the non-7 pandemic Vibrio cholerae in Vibrio Watch with this isolate VC228305 added in. This tree is shown here with the sequence type in the Octavia scheme beside the nodes of the tree. You can see that VC228305 is found in a clade with other sequence type 170 isolates. The other isolates that are clinical isolates from Mexico, so are presumably from endemic cholera disease in Mexico. It's interesting to note that VC228305 is not in the 7 pandemic lineage, so we could predict from this analysis that it is quite unlikely to cause a large epidemic. And in fact, in hindsight, this was the case for the Argentina 2005 outbreak, which was not a very large, it did not reach a large epidemic size. I will next talk to you about our future plans for Vibrio Watch. A key aim for Vibrio Watch in this summer of 2022 is to upload as many as possible Vibrio cholerae genomes that have been published in papers, with the goal of at least 3,500 genomes. We will also upload their metadata, which is the place and time of isolation, as well as the type of isolate, for example clinical or environmental, and also any phenotypic data we can collect on things such as antibiotics resistance profile, serogroup and serotype. In addition to uploading a large number of genomes, we would also like to include some extra tools in Vibrio Watch. One is a tool to predict whether an isolate is likely to be epidemic causing. This could be useful to epidemiologists who would like to know whether an isolate is likely to cause an epidemic. We plan to try to predict this based on whether the isolate belongs to the 7 pandemic lineage and whether it has the cholera toxin CTX-AB genes. Another feature that we are hoping to add soon to Vibrio Watch is to use the PopPunk software to identify Vibrio cholerae lineages. PopPunk is a software which has been successfully used among the Streptococcus pneumoniae research community to identify lineages based on genome data, and then to enable researchers to name lineages and to assign newly sequenced isolates to known lineages or to a new lineage. We are currently investigating using PopPunk for Vibrio cholerae. For example, I have built a PopPunk database here based on 23 isolates sequenced by Chun in 2009. PopPunk puts up the 7 pandemic clade isolates and all of the related isolates in the phylocore genome or PG clades into one big cluster, which makes sense because those are closely related isolates. All of the other diverged genomes that they sequenced were each put into their own PopPunk lineage. The way in which PopPunk has put the isolates into lineages makes good sense and agrees well with the manual analysis that Chun et al. Describe in their paper. In conclusion, Vibrio Watch is a free online platform for analysing Vibrio cholerae genomic data. We have included 390 Vibrio cholerae genomes so far, but aim to increase this to 3,500 by this summer and even more afterwards. Key analyses that you can do using Vibrio Watch are to analyse assembly statistics, perform MLST and core genome MLST analyses, to find related isolates using core genome MLST or phylogenetic trees, to identify virulence genes and predict antibiotic resistance. Thank you very much for listening, and I look forward to hearing any questions or comments that you have. I would like here to acknowledge the great help and work of my colleagues at the Wellcome Sanger Institute and also our collaborators who have developed Pathogen.watch at the Centre for Genomic Pathogen Surveillance, as well as John Lees at the European Bioinformatics Institute, who is helping us to implement PopPunk for Vibrio cholerae. Lastly, but very importantly, I would like to say thank you to our funders who have funded our work. These are the University of Oxford, Wellcome Trust, the Sanger Institute, and the Bill and Melinda Gates Foundation. Thank you very much.